Okay, let's unpack this. Today, we're doing a deep dive into a group of bacteria that will absolutely be central to your work as a dental professional. Streptococcus.
That's right.
Seriously, these microorganisms are fundamental players in both health and disease, and really understanding them is, well, it's critical for your career.
Exactly. And we're doing this deep dive based on a stack of sources you've shared, everything from slide presentations and PDF excerpts to detailed articles. Our goal is to synthesize all this information and make it directly relevant to you as a dental student.
Right.
We want to show how this microbiology connects explicitly to clinical dentistry.
Our mission is to go beyond just textbook definitions. We want to explore not only what Streptococcus species are, but why they matter so much in the oral cavity and actually the broader human body.
Yeah, think of this as bridging the gap between your microbiology lectures and your future practice in the operatory.
It's about understanding the bug so you can effectively fight the bite, whether that's tooth decay, endodontic infections, or, crucially, preventing those really serious systemic issues linked to the mouth.
This is foundational, practical knowledge you'll use every single day. You really will.
So we're going to start with the very basics of what these bacteria look like and how they behave. Then we'll get into the fascinating and sometimes pretty complex ways we classify them to tell different species apart. After that, we'll explore their toolkit, the virulence factors that allow them to cause harm. We'll then dive into the specific diseases they cause, paying close attention to the oral manifestations and those crucial oral systemic links.
And finally, we'll look at how labs identify them and, most importantly for clinical practice, how we treat these infections and, critically in dentistry, how we prevent some of the most serious ones.
So let's jump right in. Where do we start with getting to know these microbes?
Let's get started by getting to know these microbes on a fundamental level, their general characteristics and morphology.
All right, so general characteristics and morphology. What are the absolute first things the sources tell us about them when we look under a microscope?
Okay, the sources consistently start with their gram-stain reaction and basic shape. They are gram-positive Kochi.
Gram-positive, meaning they stain purple, right, because of that thick cell wall.
Exactly. They have a thick peptidoglycan layer in their cell wall, which holds onto the crystal violet stain. This cell wall structure is a key feature. It influences how they interact with the environment and, importantly, with antibiotics.
And Kochi just means they're round, like little spheres.
Precisely. They are spherical or sometimes slightly ovoid, kind of egg-shaped. But the truly distinctive feature, and the one that gives the genus its name, is their characteristic arrangement.
Ah, the chains.
Yes. Streptococcus typically arrange in pairs, or, more famously, in chains. The name Streptococcus itself comes from the Greek word streptos, meaning twisted chain.
A twisted chain. That image really helps. Why do they form these chains, though? Is it just random clumping?
Not at all. It's very specific. It's a direct consequence of how they undergo cell division. They divide along a single axis, like just in one direction.
Okay.
So as the cells replicate and divide, they tend to remain attached end to end rather than separating completely. That forms these linear arrangements.
Does it make sense?
And the sources point out that this chain formation is often more pronounced and easier to see clearly in liquid cultures like broths or in clinical samples like pus.
Right, where they have more space to stretch out compared to growing on a solid agar plate.
Exactly.
So gram-positive, round, and they form chains because of their division pattern. What else is notable about their basic structure or movement? Are they swimmers?
Nope. A couple of other key identifying features mentioned in the sources help differentiate them. They're largely non-motile, meaning they don't have flagella or other structures for independent movement. They pretty much rely on environmental currents or host processes for transport.
So they just go with the flow.
Basically. And critically, they are non-sporing. Unlike genera like Bacillus or Clostridium, they don't form those incredibly resistant endospores that can survive boiling or disinfectants for long periods.
Ah, okay. So no survival bunkers. That makes them a bit less hardy in the environment than spore formers?
Definitely. That lack of spore formation makes them less environmentally resistant.
Okay, so no active swimming and no hibernation capsules. How do they get their energy? What about oxygen? Are they picky?
They are classified as facultative anaerobes. This is a crucial adaptation for them.
Facultative anaerobes, meaning they can live with or without oxygen.
Exactly. It means they can perform aerobic respiration if oxygen is available. But they're perfectly capable of switching to fermentation pathways when oxygen is absent or low.
Which makes them super versatile.
Absolutely. This allows them to thrive in various environments within your body, from oxygenated tissues like the surface of the throat to the relatively low oxygen environments found in deep periodontal pockets or within abscesses.
So they're pretty flexible about oxygen. The sources also described many of the parasitic forms as fastidious. What does that imply for growing them in the lab? Are they easy to culture?
Well, being fastidious means they have complex nutritional requirements. They're picky eaters, basically. They can't just grow on simple, basic laboratory media.
Okay.
They need specific growth factors, vitamins, or amino acids that have to be provided by enriched media. So to cultivate them effectively in the lab, especially the strains isolated from patients, you typically need media supplemented with things like blood or serum.
Like blood agar or chocolate agar.
Exactly those. Those provide the necessary extra nutrients for optimal growth.
Makes sense. Picky eaters need the good stuff. When you do grow them on a plate, what do their colonies actually look like? Anything distinctive?
The sources mention that they generally form small, non-pigmented colonies on agar plates. They're often described as translucent or maybe opaque white-gray. Compared to some other bacteria that might produce, bright yellow or red pigments, Streptococcus are usually pretty understated visually.
So, small and whitish-grayish. It's a clue, maybe, but not a defining feature on its own.
Right. It's part of the picture.
And regarding their hardiness outside of a living host or that nutrient-rich lab environment, are they tough? Can they survive drying out?
Not especially, no. The sources note that Streptococcus are relatively sensitive to drying, heat, and many common disinfectants.
Now that is immediately practical knowledge for you listening. Sensitivity to drying, heat, and disinfectants directly supports the fundamental principles of infection control in your future dental practice.
Absolutely.
It explains why autoclaving instruments using that high heat and pressure, why maintaining clean, dry surfaces and using chemical disinfectants are so incredibly effective against these common oral microbes.
Exactly. Knowing this vulnerability reinforces the rationale behind rigorous sterilization and disinfection protocols. It's not just about following rules blindly. It's about understanding the biology of the target organism you're trying to eliminate.
Couldn't agree more. And speaking of fundamental lab characteristics, the sources really emphasize one key biochemical test that helps differentiate Streptococcus from another major group of gram-positive cocci you'll encounter. The catalase test.
Yes, this is probably the most basic and essential biochemical test for starting to sort out gram-positive cocci. It differentiates Streptococcus from Staphylococcus. Staphylococcus are also gram-positive cocci, but they usually arrange in clusters like grapes, not chains.
Okay, so shape is one clue, catalase is another. How does the test work?
It's simple. You take a bit of the bacterial colony and add a drop of hydrogen peroxide. Catalase-positive organisms, like Staphylococcus, produce the enzyme catalase. This enzyme breaks down hydrogen peroxide into water and oxygen.
And oxygen means bubbles.
Exactly. You see vigorous bubbling. That's a positive catalase test, pointing towards Staphylococcus.
And Streptococcus.
Streptococcus lack the catalase enzyme, so when you add hydrogen peroxide to a streptococcal colony, nothing happens. No bubbles. They are catalase negative. This is a rapid, critical first step in the lab when you encounter gram-positive coccus. No bubbles.
Right.
Think Streptococcus, or maybe Enterococcus. Bubbles. Think Staphylococcus.
Simple, but profoundly important for that initial fork in the diagnostic road. Okay, so that gives us the basic appearance, arrangement, and a key enzyme difference. But as we've hinted, the genus Streptococcus contains many, many different species. It's a huge family.
It really is.
How do microbiologists and clinicians make sense of this diversity? How do we sort them out? That brings us to classification.
Right. Classification is absolutely necessary because different streptococcal species cause vastly different diseases, live in different places, and crucially, require different approaches to diagnosis and treatment. The sources lay out multiple interconnected methods used for classification. It's not just one system.
And these methods range from things you can see on a plate to their genetics.
Exactly. The primary methods mentioned are based on their hemolytic activity on blood agar, what they do to red blood cells. Then there are their serological properties, specifically the famous Lancefield grouping, based on cell wall antigens. Then you have biochemical and physiological characteristics, like the catalase test we just discussed, and others. Their basic morphology and arrangement gives clues, too. And increasingly, molecular methods looking at their DNA or RNA are used for finer distinctions.
Okay, let's start with the one that gives a really clear visual. Hemolytic activity on blood agar. The sources show us 3 distinct patterns. What are they?
Right. When you grow Streptococcus on a blood agar plate, which is basically nutrient agar mixed with sheep's blood, you observe how they interact with the red blood cells right around their colonies. First, there's alpha hemolysis.
Alpha hemolysis, what does that look like?
This isn't complete destruction of the red blood cells. Instead, you see a partial lysis that results in a greenish discoloration of the agar surrounding the colony. It's kind of a murky green zone.
And the sources explain that green color is due to the production of hydrogen peroxide by the bacteria, which then oxidizes the hemoglobin in the red blood cells to methemoglobin, giving that characteristic greenish hue. So it's an incomplete breakdown, an alteration.
Correct. Key examples of alpha hemolytic streptococci, and ones highly relevant to you in dentistry, are the viridin streptococci. That's a whole diverse group found commonly in the oral cavity, and Streptococcus pneumonia, often just called pneumococcus. The image in the sources clearly shows that greenish zone of alpha hemolysis.
Got it. Alpha, partial, greenish zone. Next up is beta. Velothesis. This looks much more dramatic in the pictures.
It does. Beta hemolysis results in a wide, clear, transparent zone around the colony. The red blood cells in the agar in that zone have been completely lysed, totally destroyed, leaving a clear halo where you can practically see through the agar.
And that complete lysis is caused by potent bacterial toxins called hemolysins, specifically streptolysin O and streptolysin S, as the sources detail. They just shred the red blood cells.
Precisely. And the sources are very clear that most of the really pathogenic streptococci that cause widespread serious infections, especially Streptococcus pyogenes, the notorious group A strep that causes strep throat, are beta hemolytic. The image vividly illustrates that clean, clear zone around the beta hemolytic growth.
So alpha is partial green, beta is complete clear. In the third category, what's gamma?
Gamma, chemolysis. This isn't really hemolysis at all, which can be a bit confusing nomenclature-wise. It simply means there's no observable effect on the red blood cells around the colony. No lysis, no clearing, no discoloration. The agar just stays red right up to the edge of the colony.
The image shows that perfectly just the bacterial growth sitting on the red agar, no change around it. And the sources mention Enterococcus faecalis as a prominent example in this category.
Yes, and this is an important historical and dental point you need to remember. Enterococcus faecalis was originally classified within the Streptococcus genus, specifically as part of Lancefield group D, and was often referred to as Streptococcus faecalis.
Ah, I see.
It's now recognized as belonging to a separate genus, Enterococcus, based on genetic and other differences. But it's typically non-hemolytic or gamma hemolytic on the blood agar, and as we'll definitely discuss later, it's a highly significant pathogen in persistent endodontic infections. So that gamma hemolysis pattern is still relevant.
Okay, so hemolysis on blood agar gives us one way to broadly categorize them: alpha, beta, or gamma. Then there's the system based on serology, the Lancefield grouping. Tell us about that.
This system, developed by the brilliant Rebecca Lancefield back in the early 20th century, is based on identifying specific heat-stable carbohydrate antigens, often called the C substance or C carbohydrate, present in the cell wall of many streptococci.
So it's looking for specific tags on the bacteria's surface?
Essentially, yes. It's a serological method, meaning it uses antibodies to detect these specific antigens. It's a very useful system, particularly for classifying the beta hemolytic strains, although some alpha and gamma hemolytic strains also possess these antigens, like the original group E streptococci, including the Enterococci we just mentioned.
The sources mention that beta hemolytic strains are divided into groups A through U, something like 21 different groups identified based on these carbohydrate antigens. But they highlight the groups most commonly associated with human infections.
Yes, while there are many groups, the key ones causing human diseases are A, B, C, D, and G. The sources detail the most important species linked to these groups. Group A is Streptococcus pyogenes. This is arguably the most famous Streptococcus, a major human pathogen responsible for things like strep throat, scarlet fever, skin infections, and those dangerous post-infectious sequelae like rheumatic fever.
Group A is the big one.
Then Group B is Streptococcus agalaxiae. This one is primarily known for causing serious infections like sepsis and meningitis in newborns, transmitted from the mother during birth.
Okay, Group B and newborns.
Group D historically included both the Enterococcus species, like the dentally relevant Enterococcus faecalis, and non-enterococcal species like Streptococcus bovis. The sources correctly point out that Enterococcus is now its own genus, but its historical link to Group D antigen is important context, and S. bovis group organisms are still considered Group D strep.
And groups C and G.
Group C and G are also associated with human infections, often causing diseases similar to Group A, like pharyngitis or skin infections, but they tend to be less common.
So Lancefield grouping is really essential for identifying major systemic pathogens like S. pyogenes and S. agalactiae. But the sources make a really critical distinction here that's vital for you as a dental student.
Yes, this is where it gets particularly relevant for your focus. The sources explicitly state that many of the Streptococci found as normal inhabitants of the oral cavity, particularly the alpha-hemolytic viridin Streptococci, do not possess these specific Lancefield group-specific carbohydrate antigens.
Ah, okay. So the primary classification method we use for many of the most dangerous systemic strep infections isn't applicable to a large number of the strep species living right there in our mouths, the ones you'll be dealing with day in and day out.
Exactly. You cannot use Lancefield grouping to classify most oral Streptococci, like the mutans group or the mitis group. This really underscores why other methods, particularly biochemical tests and more recently molecular techniques, are absolutely essential for identifying and classifying the species you'll encounter most frequently in dental settings. Lancefield is great, but it doesn't cover everything, especially not the oral guys.
Okay, that leads us perfectly into those biochemical or physiological properties. These are the specific lab tests used to tell different species apart based on what they metabolize or what enzymes they produce, especially when Lancefield grouping isn't an option or needs confirmation.
Right. We already covered the cornerstone, the catalase test, unequivocally negative for all Streptococcus and Enterococcus.
Got it. No bubbles.
Beyond that, other key tests detailed in the sources include sugar fermentation tests. By seeing which specific sugars a bacterial isolate can ferment, meaning break down anaerobically to produce acid, you can differentiate species. Different strep species have distinct sugar fermentation profiles, and this is particularly useful for sorting out the various viridin species found in the mouth.
So feeding them different sugars and seeing if they make acid, what about tests that help pinpoint specific important pathogens quickly?
There are several key ones used routinely. Bacitracin sensitivity, this is a classic. Streptococcus pyogenes, group A, is uniquely sensitive to a very low concentration of the antibiotic bacitracin, while most other beta-hemolytic streptococci are resistant.
How is that used in practice?
Well, if you have a beta-hemolytic colony growing, say from a throat swab, you place a small paper disc containing bacitracin onto the agar plate where it's growing. If you see a clear zone of inhibition, no growth, around that disc after incubation, it's a strong presumptive identification for S. pyogenes. It's a quick screen for group A strep.
Okay, that's a neat trick for potentially identifying group A strep fast. Now, what about Streptococcus pneumonia? It's alpha-hemolytic, like the viridins group, and often lacks Lancefield antigens. How do we reliably differentiate pneumococcus from all those oral viridin strep, since that distinction is clinically very important?
This is where optogen sensitivity and bile solubility come in. These are the two crucial classic tests used to distinguish S. pneumonia from the viridins group Streptococci.
Optogen sensitivity. What's optogen?
Optogen is a chemical ethyl hydrocuprin hydrochloride. Streptococcus pneumonia is uniquely sensitive to it. So, similar to the bacitracin test, you place an optogen-impregnated disc on a culture plate inoculated with the alpha-hemolytic isolate. S. pneumonia will show a distinct zone of inhibition, no growth, around the disc. Viridin streptococci, on the other hand, will grow right up to the edge of the disc because they are resistant.
So optogen sensitivity is a key signature for pneumococcus among the alpha-hemolytics, and bile solubility. What's that about?
Streptococcus pneumonia has an autolytic enzyme system that gets activated by bile salts, like sodium deoxycholate. So if you make a suspension of the bacteria in saline and add a bile salt solution, typically 10%, the pneumococcal cells will lyse or dissolve. The cloudy bacterial suspension will become clear.
They just dissolve in bile.
Pretty much. Other alpha-hemolytic streptococci, like the viridins group, lack this bile-activated autolysin, so they are not soluble in bile, and the suspension remains turbid or cloudy.
Wow. So if you find an alpha-hemolytic gram-positive cocci that is sensitive to optogen and is soluble in bile, you can be very confident it's Streptococcus pneumonia.
Extremely confident. Those two tests together are really definitive for identifying pneumococcus in the clinical lab.
Okay, those are critical for pneumococcus. Any other important biochemical tests mentioned for other groups?
Yes, the sources mention the PYR test. This tests for the enzyme pyrrolidinol arylamidase. It's positive, usually indicated by a color change to red or pink after adding a reagent for group A Streptococcus pyogenes and also for Enterococcus species.
So PYR is another check for group A, and it also helps identify Enterococci.
Correct. Then there's the CMP test.
Yeah.
You mentioned group B strep agalactiae before. The CMP test is specifically used to identify group B. It relies on an interesting interaction.
How does it work?
You streak the unknown Streptococcus perpendicular to, but not touching, a streak of a specific strain of Staphylococcus aureus that produces a toxin called beta-lysine. Group B strep produce a protein called the CAMP factor. Where the TMP factor diffuses and meets the beta-lysine from the staph, there's a synergistic enhanced zone of beta hemolysis, often forming a characteristic arrowhead shape pointing towards the staph streak. The sources show a great image of this.
That arrowhead image is really distinctive. So CMP test points to group B. What about the bile esculin test, or BEA?
Yes. The BEA test is primarily used for identifying Enterococci and also some other group D strep like the S. bovis group. These organisms can grow in the presence of bile, which inhibits many other bacteria, and they can hydrolyze a compound called esculin. When esculin is hydrolyzed, one of the breakdown products reacts with ferric citrate in the medium to produce a distinct black precipitate.
So if the tube or slant turns black, it suggests Enterococcus or maybe group D strep.
Exactly. And the sources also reiterate those remarkable physiological traits of Enterococci that serve as identifying features and contribute to their resilience. Remember, they can grow in really challenging conditions. High salt concentration, 6.5% ACL broth, high bile concentration, 40%, a wide pH range including alkaline conditions, pH 9.6, and they can even survive heating at 60 degrees C for 30 minutes.
That list really drives home why Enterococcus faecalis is such a problem in things like failed root canals. It's just incredibly tough and built to survive harsh environments that would kill off many other bacteria.
Precisely. That inherent toughness is a key part of its profile, bringing it back to that direct dental relevance.
And morphology. Does the shape ever help beyond just chains versus pairs?
Well, the sources do loop back to morphology as a classification aid sometimes. For instance, they mention the specific lanceolate or lancet-shaped pointed appearance of the Diplococci pairs of Streptococcus pneumonia. Seeing those distinctive shapes under the microscope can be a helpful visual clue.
So a slightly pointed pair might suggest pneumococcus. Good to know. And finally, the most modern method mentioned, molecular classification. How does that fit in?
This is where things are really advancing. Molecular classification involves looking directly at the bacteria's genetic material. Techniques like sequencing specific genes, often the 16S ribosomal RNA gene, which is present in all bacteria but varies slightly between species, or doing broader DNA homology studies allow us to determine how related different strains of the species are at a genetic level.
So it's like bacterial DNA fingerprinting or ancestry testing?
Kind of, yeah. It provides a more objective and arguably more stable classification system compared to relying solely on phenotypic traits like sugar fermentation or hemolysis, which can sometimes be variable or change depending on growth conditions.
And is this particularly important for any specific group of strep?
The sources note that this is particularly important for complex and rapidly evolving groups, like the oral Streptococci. Molecular techniques are constantly refining our understanding of the species relationships within the viridans group, for example, sometimes leading to changes in nomenclature or the definition of species. It's an active area of research, especially for the bugs in your neighborhood, the mouth.
Okay, so using a combination of what they do to blood cells, hemolysis, their cell wall tags, Lancefield, what they eat and what enzymes they make, biochemicals, their shape, and now their DNA helps us navigate this incredibly diverse genus. And the sources really helpfully nail down the key oral Streptococci groups you'll need to know as a dental student based on all this classification.
Yes, they explicitly list the groups residing in or causing significant issues in the mouth, which you should definitely familiarize yourself with. First, the mutans group, starring S. mutans and S. sabrinus. These are identified unequivocally as the major agents of dental caries. Think cavities, think mutans group.
Got it. Mutans, caries.
Then, the salivarius group, like S. salivarius and S. vestibularis. These are very common commensals, often found predominantly on the tongue and in saliva.
Okay, salivarius group, mostly tongue dwellers.
The anginosus group, which includes S. consulatus, S. intermedius, and S. anginosus. This group is sometimes also called the S. milleri group. They're important because they are frequently implicated in pyogenic, pus-forming infections, including dental alveolar abscesses and endodontic infections. They often like low oxygen or added CO2.
Anginosus Miller group, abscesses, endo-infections.
Then there's the mites group. This is another large group found in dental plaque and saliva, including species like S. sanguinis, S. gordonii, S. oralis, and S. parasanguinis. They're associated with early plaque formation, sometimes caries, but also significantly with infective endocarditis, especially S. sanguinis.
Mitis group, plaque, endocarditis risk.
And finally, they mentioned anaerobic streptococci. These were formerly classified in a separate genus called Streptococcus, but many are now back in Streptococcus. They're often found in mixed anaerobic infections like pericoronitis, infection around a partially erupted wisdom tooth, and other dental-alveolar abscesses.
Anaerobic strep equals mixed infections, abscesses. That grouping is incredibly helpful. It takes the complexity of all those oral strep species and organizes them into clinically relevant categories based on where they tend to live and, more importantly, the specific problems they tend to cause in the dental realm. Knowing these specific groups is really a game changer for understanding oral pathology.
Absolutely. It moves you from just abstract microbiology concepts to understanding the specific bacterial players driving the specific oral diseases you're going to be diagnosing and treating.
Okay, so we have a much better handle on what they are and how we classify them. Now let's really get into the nitty-gritty of how they actually make us sick. That requires exploring their virulence factors, the specific tools, molecules, and strategies these bacteria use to adhere to our tissues, evade our powerful immune system, and cause damage.
Right. Virulence factors are essentially the weapons and armor that enable these bacteria to transition from just being present to actually causing disease in a host. They're a crucial part of that dynamic interaction between the bacterium and the human body. The sources provide a really detailed look at the virulence factors of several key species.
And they start with the quite extensive arsenal of Streptococcus pyogenes, group A strep, GAS. Even though it's often thought of as causing strep throat or skin infections, understanding its virulence factors provides a great template for how many streptococcus operate.
Correct. For S. pyogenes, the M protein is highlighted consistently as a major, perhaps the most important, virulence factor. It's a fibrillar protein that sticks out from the cell surface, kind of like hair.
And it does multiple things, right? It helps it stick.
Yes, its functions are critical. Firstly, it acts as an adhesin, helping the bacteria bind specifically to host epithelial cells in the pharynx and on the skin. That binding is the essential first step in establishing an infection. But perhaps even more importantly, M protein is a potent antiphagocytic factor.
Antiphagocytic.
Yeah.
Meaning it helps them avoid being eaten and destroyed by our immune cells, like neutrophils and macrophages. That sounds crucial for survival.
Absolutely crucial. The sources specifically note that M protein achieves this partly by binding to fibrinogen from our plasma, creating a coat, and also by binding to and inactivating C3b. C3b is a key component of the complement system, one of our innate immune defenses. C3b normally acts as an opsonid, coats bacteria, essentially tagging them for destruction by phagocytic cells.
Like putting a kick me sign on the bacteria for the immune cells.
Exactly. By inactivating C3b, the M protein prevents this tagging, allowing the bacteria to effectively evade being engulfed and killed by phagocytes. It's a major immune evasion strategy. Additionally, the M protein is highly variable. Antigenically, there are over 100 different M types, which helps the bacteria evade host antibody responses, and this variation is used for serotyping S. pyogenes.
So the M protein helps them stick and hide from the cops, the phagocytes. That's a powerful combination. What else is part of the S. pyogenes toolkit for protection?
They also possess a capsule made of hyaluronic acid. Now, what's interesting about this capsule is that hyaluronic acid is also a major component of human connective tissue.
Molecular mimicry. Self.
Exactly. This makes the capsule non-antigenic, or very poorly antigenic. The host immune system doesn't readily recognize it as foreign. This camouflage helps the bacteria blend in and avoid immune detection to some extent. The capsule also acts as a physical barrier, contributing weakly to antiphagocytosis and also helping prevent the bacteria from drying out.
A kind of invisibility cloak made of host material. Clever.
Yes. And there are other adherence factors too. The sources mention lipoteichoic acid, LTA, and the F protein, also called protein F, or SFBI. These are components found in the bacterial cell wall that specifically bind to fibronectin, a protein commonly found on the surface of host epithelial cells. This binding provides another way for the bacteria to securely attach to tissues, reinforcing the initial adherence mediated by M protein.
So multiple ways to make sure they stick where they want to be. Once they're stuck, how do they spread? Do they just sit there?
No, they often spread. S. pyogenes produces several enzymes that act as spreading factors, allowing them to break through tissue barriers. One major one is streptokinase, also known as fibrinolysin.
Fibrinolysin. Sounds like it breaks down fibrin.
That's exactly what it does, indirectly. Streptokinase activates plasminogen, a host enzyme precursor, converting it into active plasmin. Plasmin is the body's natural clot buster. It degrades fibrin, the protein meshwork that forms blood clots. By activating plasmin, streptokinase allows the bacteria to dissolve the fibrin barriers that the host's immune system tries to build to wall off an infection. This facilitates the spread of the bacteria through tissues.
So they literally dissolve the fences the body tries to put up around them. Pretty strategic for invasion.
Very much so. Then there's hyaluronidase. We just talked about the hyaluronic acid capsule. Hyaluronic acid is also a major component of the extracellular matrix, the glue that holds cells together in connective tissues. Hyaluronidase is an enzyme that breaks down this hyaluronic acid.
So it dissolves the tissue glue.
Essentially, yes. By degrading this ground substance, hyaluronidase helps the bacteria penetrate deeper into tissues and spread more easily. It's like paving the way for invasion.
Another enzyme designed for breaking down host tissue components. What else?
And DNases, sometimes collectively called streptococcus, these enzymes hydrolyze DNA. Now, why is that useful for the bacteria?
Maybe from dying cells, like in pus.
Exactly. When our immune cells, particularly neutrophils, fight infection, they release their DNA to form structures called neutrophil extracellular traps, or nets, which are sticky webs designed to trap bacteria. Also, dying host cells and bacteria release DNA, contributing to the thick, viscous nature of pus. DNases break down this extracellular DNA.
And that would make the pus thinner.
Yes, it reduces the viscosity of pus and liquefies exudates, making it easier for the bacteria to move around within an abscess and spread further. It helps them escape being trapped.
Gross, but clearly an effective strategy for dissemination. They also seem to have another trick for messing with the immune system's response. The sources mention something called C5a peptidase.
Yes, this is another targeted immune evasion mechanism. C5a is a potent chemoattractant protein generated by the complement system. Its job is to signal and recruit phagocytic cells, like neutrophils and macrophages. To the site of infection, it's like sounding the alarm bell.
Okay.
C5a peptidase is an enzyme produced by S. pyogenes that specifically cleaves and inactivates C5a. By destroying this come-here signal, the bacteria significantly reduce the influx of neutrophils and macrophages to the infection site, further enhancing their ability to evade phagocytosis and establish themselves.
Wow, so they prevent the immune reinforcements from even arriving effectively, and they have the M protein to protect them from the ones that do get there. That's quite the multi-layered defense system. And then on top of all that, there are the powerful toxins.
The toxins are really responsible for some of the most severe systemic symptoms and tissue damage associated with S. pyogenes infections. The sources highlight the streptococcal pyrogenic exotoxins, SPEs, sometimes referred to as erythrogenic toxins, because they cause the rash of scarlet fever. There are several types like SPEA, B, C, F. These are famously classified as superantigens.
Superantigens. What makes them super entire compared to regular antigens or toxins? How do they work?
It's about how they interact with the immune system, specifically T cells. Normal antigens bind to the T cell receptor and the major histocompatibility complex, MHC class II molecule, on antigen-presenting cells in a very specific groove, activating only a tiny fraction of T cells that recognize that specific antigen.
Okay, very targeted activation.
Superantigens, however, don't bind in that specific groove. They bind to the outside surfaces of the T cell receptor and the MHC class II molecule, essentially cross-linking them in a non-specific way. This abnormal binding triggers the activation of a huge number of T cells, maybe up to 20% or more of the body's entire T cell population, compared to less than 0.01% for a normal antigen.
A massive, non-specific activation. What's the consequence of activating so many T cells at once?
The consequence is a massive, uncontrolled release of pro-inflammatory cytokines, things like tumor necrosis, factor alpha, TNF4, interleukin-1, IL-1, interleukin-2, IL-2. This huge flood of cytokines is often called a cytokine storm. It causes widespread systemic effects like high fever, diffuse rash like in scarlet fever, low blood pressure leading to shock, and potentially multi-organ failure. Superantigens are directly implicated in the pathogenesis of streptococcal toxic shock syndrome, STSS, and also contribute significantly to the extensive tissue destruction seen in severe invasive infections like necrotizing fasciitis.
Wow, so they basically hijack and massively overstimulate the host's own immune system, turning it against itself to cause widespread damage. That really explains their role in those terrifying, rapidly progressive diseases.
It really does. And finally, let's not forget the hemolysins we discussed earlier as causing beta hemolysis. Streptolysin O, SLO, and streptolysin S, SLS. These are also considered important toxins and virulence factors.
Because they do more than just pop red blood cells.
Yes. While their ability to lyse red blood cells is their defining feature on blood agar, the sources also note they can lyse other host cells, including white blood cells, like neutrophils and macrophages, which contributes to tissue damage and helps the bacteria evade immune clearance. They can also lyse platelets. Streptolysin O specifically is mentioned as having cardiotoxic activity, meaning it can potentially damage heart muscle cells. And SLO is the toxin that stimulates the production of antibodies measured by the ASO, anti-streptolysin O, titer test, which is used diagnostically. SLS is stable in oxygen and responsible for the surface hemolysis you see, while SLO is oxygen labile.
That is a truly formidable set of tools for S. pyogenes adherence factors, immune evasion proteins, spreading enzymes, superantigens, and cell-lysing toxins. Okay, what about virulence factors for other key species? You mentioned Streptococcus pneumonia. What's its main weapon?
For Streptococcus pneumonia, pneumococcus, the single most important virulence factor highlighted across the sources is its thick polysaccharide capsule.
The capsule again. Why is it so critical for pneumococcus?
Similar to the S. pyogenes capsule, its primary role is defense against phagocytosis. It's a slippery protective layer that prevents immune cells, especially neutrophils, from easily engulfing the bacterium, particularly in the absence of specific antibodies directed against that capsular type. The host immune system really struggles to clear encapsulated bacteria without those specific antibodies.
So the capsule is its shield.
Absolutely. This capsule is so crucial to its virulence that the different types of capsular polysaccharides, and there are over 100 different serotypes based on these capsule variations, form the basis for pneumococcal vaccines. It was also the capsular material that was central to Griffith's famous transformation experiment back in 1928, which first demonstrated bacterial transformation. Other virulence factors listed for pneumococcus include hemolysins, like nimlysin, which is a pore-forming toxin that damages host cells, leukocidin, a toxin that specifically kills white blood cells, and an IgA1 protease.
IgA1 protease, what does that do?
Secretory IgA, SIGA, is an important antibody found on mucosal surfaces, like in the respiratory tract, where it helps prevent bacteria from attaching. IgA1 protease is an enzyme produced by pneumococcus that specifically cleaves human IgA1 antibodies, helping the bacteria to colonize respiratory surfaces more effectively.
So it disables the first line of antibody defense on the mucosal surface. Clever. So for pneumococcus, the capsule is really king for causing invasive disease aided by toxins in that protease. What about the hardy Enterococci? What are their main virulence factors?
For Enterococcus species, like E. faecalis, their virulence seems to be less about producing potent secreted toxins or spreading enzymes like S. pyogenes, and more about their inherent ability to survive and persist in really harsh environments, their capacity to form biofilms, and their significant intrinsic and acquired antibiotic resistance.
So their toughness is their virulence.
In many ways, yes. The sources list their documented heat resistance, their ability to grow in high salt concentrations, and the presence of bile, their tolerance of a wide pH range, and their intrinsic resistance to certain classes of antibiotics, like cephalosporins and aminoglycosides at low levels, as key traits that contribute to their pathogenicity. These characteristics allow them to survive in hospital environments, colonize patients, and persist in sites like the urinary tract or inside root canals where conditions are challenging. They're simply very difficult organisms to eradicate.
That persistence and resistance really defines them as opportunistic pathogens, enabling them to cause chronic or difficult-to-treat infections, especially nosocomial ones. And finally, let's pinpoint the key virulence factor for S. mutans, the primary culprit in cavities.
For Streptococcus mutans and its close relatives in the mutans group, the sources consistently highlight the formation of biofilms, which we commonly know as dental plaque, as their fundamental virulence mechanism in the context of dental caries.
That's it. It's not just about making acid. It's about making acid within that sticky, organized community on the tooth surface. That ability to form and thrive in a biofilm is the key.
Exactly. It's a combination of factors enabled by the biofilm lifestyle. First, they produce those extracellular polysaccharides, glucans and fructans, from sucrose, which allows them to adhere tenaciously to the tooth surface and to each other, building up the plaque matrix. Second, this biofilm structure provides a protective microenvironment, shielding the bacteria from saliva flow, buffering agents, and even antimicrobial agents to some extent. Third, it allows for incredibly close contact between the bacteria and the tooth surface. And fourth, it enables the bacteria to concentrate their metabolic activity and the resulting acid production right against the enamel surface, creating a localized, intensely acidic microenvironment that drives demineralization.
So it's the sticking power, the acid production, acidogenicity, the ability to survive that acidoricity, all happening within that protective, organized biofilm structure. That's what makes them such effective carries pathogens.
Precisely. It's the whole package, orchestrated within the biofilm.
Understanding these virulence factors, the specific strategies like M protein, capsules, enzymes, toxins, biofilm formation, really demystifies how these bacteria transition from just being present in or on our bodies to actively causing disease. Okay, let's move on to the direct result of this interaction. The pathogenesis and the wide range of diseases these different Streptococci cause.
Right, we need to cover the spectrum from the major systemic infections caused by some species, and then really focus in on those with direct and critical relevance to you in dentistry.
Let's start again with the diseases caused by the heavily armed Streptococcus pyogenes, Group A strep, GS. Where does it come from and who gets sick?
The sources emphasize that humans are the only natural reservoir for S. pyogenes. There's no animal source. Transmission is mainly through direct contact with secretions, like saliva or nasal discharge, from an infected person, via respiratory droplets produced during coughing or sneezing, or sometimes through contaminated food or fomites, inanimate objects that carry the bacteria.
And how does it get in?
The bacteria typically enter the body through breaks in the skin or by colonizing the mucous membranes of the pharynx, the throat. While anyone can potentially get these infections, the sources note that children, particularly school-aged children, are especially susceptible to pharyngeal infections, strep throat, and certain skin infections like impetigo.
And the diseases caused by GAS are broadly classified into two types, right? Suppurative and non-suppurative. What's the difference?
Right. Suppurative infections are those characterized by the formation of pus. These are due to the direct invasion, multiplication, and spread of the bacteria within tissues, causing inflammation and damage.
What are some classic examples of suppurative GS infections?
The most common are respiratory infections like streptococcal pharyngitis, classic strep throat, and tonsillitis. If the infecting strain produces one of those pyrogenic exotoxins, superantigens, the pharyngitis can be accompanied by scarlet fever, characterized by that distinctive sandpaper-like rash, flushed face with paleness around the mouth, and a strawberry tongue.
Okay, throat and scarlet fever. What about skin?
Skin infections are also very common. These include impetigo, which are those superficial crusting sores often seen in children, erysipelas, a distinct type of superficial cellulitis with raised, sharply demarcated borders, often on the face or legs, and deeper cellulitis, which is infection spreading through the subcutaneous tissues.
And sometimes it gets much worse, spreading deeper.
Yes, more severe invasive suppurative infections can occur if the bacteria get into the bloodstream or deeper tissues. Examples listed include septicemia, bacteria multiplying in the bloodstream, osteomyelitis, infection of the bone, puerperal sepsis, a historically significant infection of the uterus after childbirth, also called childbed fever, and the devastating necrotizing fasciitis.
Necrotizing fasciitis. That's the flesh-eating bacteria infection we hear about.
That's the one, though the term flesh-eating is a bit sensationalized. S. pyogenes is a major cause. It involves rapid, extensive destruction of the soft tissues, muscle, fascia, and subcutaneous fat. It's often associated with strains producing high levels of toxins and spreading factors, particularly those superantigens, leading to severe systemic illness like streptococcal toxic shock syndrome, along with the local tissue destruction. It's a true surgical emergency.
Terrifying. So these are all active infections where the bacteria are present and causing direct damage through invasion and toxins. Now, what about the non-suppurative sequelae? How are they different?
These are fundamentally different. They are post-infectious complications, meaning they develop after the initial streptococcal infection, usually pharyngitis, has resolved, typically weeks later. They are not caused by direct bacterial invasion or pus formation at the site of the sequelae. Instead, they are thought to be primarily immunological in nature. They occur in a relatively small percentage of individuals, usually following untreated or inadequately treated GAS infections.
So the infection is gone, but the body's reaction causes problems later.
That's the idea. The two major non-suppurative sequelae discussed are acute rheumatic fever, ARF, and acute post-streptococcal glomerulonephritis, APSGN.
Acute rheumatic fever. This is the one that particularly concerns us because of its potential long-term impact on the heart, right?
It absolutely is. ARF is primarily an inflammatory disease that can affect multiple systems, notably the joints, heart, brain, and skin. The sources detail the classic symptoms using the Jones criteria. Migratory polyarthritis, painful swelling in large joints that seems to move from one joint to another. Carditis, inflammation of the heart muscle lining or valves, which is the most serious manifestation because it can lead to permanent damage to the heart valves. Rheumatic heart disease causing leaky or narrowed valves, irregular heartbeats, and potentially heart failure later in life. Siddons, chorea, involuntary jerky movements, sometimes called St. Vitus's dance, subcutaneous nodules, painless lumps under the skin, and erythema marginatum, a distinctive non-itchy skin rash with red borders.
How does a throat infection lead to heart valve damage weeks later? What's the proposed mechanism?
The leading hypothesis is molecular mimicry causing an autoimmune reaction. The idea is that antibodies produced by the patient against certain components of the S. pyogenes bacteria, particularly the M protein, cross-react with similar-looking protein antigens found in the host's own tissues, specifically in the heart valves, joints, and brain. So the immune system, trying to fight the lingering effects of the strep infection, mistakenly attacks the body's own tissues, causing inflammation and damage.
So the body's immune response gets confused and attacks self-tissues because they resemble parts of the bacteria. That's fascinating and scary. And acute post-streptococcal glomerulonephritis, or APSGN, affects the kidneys.
Yes, this is an immune-mediated kidney disorder characterized by inflammation of the glomeruli, which are the tiny filtering units within the kidneys. It typically develops about 1-2 weeks after a GAS pharyngitis, or perhaps more commonly, 3 to 6 weeks after a GAS skin infection, pyoderma, and only certain nephrogenic strains of GAS are strongly associated with it.
What are the symptoms of APSGN?
Symptoms listed include hematuria, blood in the urine, which might make it look tea-colored or smoky, proteinuria, protein spilling into the urine, hypertension, high blood pressure, reduced kidney function, oliguria, meaning decreased urine output, and edema, swelling, which is often most noticeable around the eyes, periorbital edema, and in the ankles.
And how does this happen immunologically? Is it also cross-reaction?
This one is thought to be primarily caused by the deposition of immune complexes in the glomeruli. These complexes consist of streptococcal antigens bound to antibodies produced by the host. These antigen-antibody complexes get trapped in the glomerular filters, trigger activation of the complement system, and attract inflammatory cells, leading to damage and inflammation of the glomeruli. Unlike rheumatic fever, APSGN usually resolves completely, especially in children, although some adults may have persistent kidney issues.
These non-suppurative sequelae really underscore the critical importance of properly diagnosing and treating GAS infections, especially strep throat, with appropriate antibiotics like penicillin. Preventing these serious delayed complications is a major reason why we treat strep throat so diligently.
Absolutely. Prompt and complete treatment of GAS pharyngitis dramatically reduces the risk of developing acute rheumatic fever. The link with preventing APSGN is less clear-cut, but treatment is still essential.
Okay, let's move on from GAS to Streptococcus pneumonia, the pneumococcus. What are the main diseases it causes?
Pneumococcus is a major human pathogen, particularly known as the most common cause of community-acquired bacterial pneumonia worldwide.
Who is most at risk for pneumococcal pneumonia?
The sources consistently note the susceptible groups include infants and young children whose immune systems are still developing, the elderly whose immune systems may be waning, and individuals of any age with underlying medical conditions. These conditions include chronic lung diseases like COPD or asthma, chronic heart disease, diabetes, sickle cell disease, alcoholism, conditions that weaken the immune system like HIV/AIDS, cancer, or immunosuppressive therapy, and even preceding viral respiratory infections like influenza, which can damage the airways and make them more susceptible to secondary bacterial invasion by pneumococcus.
How does it cause pneumonia? Does it just invade the lungs directly?
It usually starts with colonization of the nasopharynx, the area at the back of the nose and throat, where it can live harmlessly in many people. Disease often occurs when the bacteria are aspirated from the nasopharynx down into the lower airways and lungs, especially if host defenses are impaired. Once in the lungs, they multiply rapidly, triggering a strong inflammatory response. Fluid and immune cells rush into the alveoli, air sacs, leading to consolidation, the filling of lung tissue, which impairs gas exchange and causes the symptoms of pneumonia: cough, fever, chest pain, shortness of breath.
So it takes advantage of weakened defenses to get into the lungs, and pneumonia is its main claim to fame, but it causes other serious infections too, right?
Yes, absolutely. Pneumococcus is also the leading cause of bacterial meningitis in adults in many parts of the world. It can spread to the meninges, the membranes surrounding the brain and spinal cord, via the bloodstream, or sometimes directly from a nearby infection site like the sinuses or middle ear.
Meningitis is incredibly serious. What else?
It's also a very frequent cause of otitis media, middle ear infections, in young children. The sources specifically mention it can gain access to the middle ear space from the nasopharynx via the eustachian tube. Other common infections caused by pneumococcus include sinusitis, infection of the paranasal sinuses, bacteremia, presence of bacteria in the bloodstream, which can occur with pneumonia or meningitis or sometimes on its own. And it can occasionally cause other invasive infections like septic arthritis, joint infection, or endocarditis, infection of the heart valves, though it's less common for endocarditis than viridans, strep, or staph aureus.
So pneumococcus is a major player in causing infections throughout the respiratory tract, ears, sinuses, bloodstream, and central nervous system. Really versatile and dangerous.
Yeah.
What about group B strep, Streptococcus agalactia? We mentioned newborns.
Yes, Streptococcus agalactia, GBS, is most famously associated with causing severe life-threatening infections in newborns, particularly within the first week of life, early onset disease, or up to several months of age, late onset disease.
What kind of infections?
The most common and serious are neonatal septicemia, bloodstream infection, and meningitis. Pneumonia can also occur. GBS is part of the normal vaginal and gastrointestinal flora in a significant percentage of healthy women, maybe 15-30%. Transmission to the infant typically occurs during labor and delivery if the mother is colonized.
That sounds preventable if we know the mother is colonized.
It is, largely. Routine screening of pregnant women for GBS colonization, usually via vaginal rectal swabs late in pregnancy, and administering intravenous prophylactic antibiotics, typically penicillin, to colonize women during labor, has dramatically reduced the incidence of early-onset neonatal GBS disease in countries where this is standard practice.
That's a huge public health success story. Does GBS cause disease in adults, too?
Yes, it can. While less common than in neonates, GBS can cause infections in adults, particularly pregnant women, causing UTIs, womb infections, and non-pregnant adults, especially the elderly or those with significant underlying medical conditions like diabetes or cancer. Infections in adults can include skin and soft tissue infections, pneumonia, bone and joint infections, UTIs, and bloodstream infections.
Okay. And Enterococcus species. We've touched on their hardiness and endodontic relevance. What's their disease spectrum?
Enterococci, especially the two main species, Enterococcus faecalis and Enterococcus faecium, are primarily opportunistic pathogens. They are part of the normal gut flora, but can cause infections when they spread to other sites, particularly in compromised hosts. They are a very common cause of urinary tract infections, UTIs, especially those acquired in hospitals or associated with urinary catheters.
UTIs are a big one for Enterococcus. What else?
They are also a frequent cause of endocarditis, typically occurring in patients with pre-existing heart conditions, prosthetic heart valves, or following procedures that cause bacteremia. Enterococcal endocarditis can be particularly difficult to treat due to antibiotic resistance. They can also cause intra-abdominal and pelvic infections, often following surgery or trauma, and wound infections, especially on hospitalized patients.
And crucially for you listening, the dental connection.
Yes, the sources specifically highlight their significant role in endodontic infections. Enterococcus faecalis is particularly notorious for being found in persistent or failed root canal treatments. Its ability to survive the harsh conditions within a root canal system, form biofilms, and resist common intracanalin medicaments makes it a major challenge in endodontics. And broadly, enterococci are major culprits in nosocomial hospital-acquired infections overall, due to their inherent resistance, ability to acquire further resistance, and their knack for surviving on environmental surfaces in healthcare settings.
Okay, that covers the major systemic players. Now let's really focus heavily on the groups most relevant to the dental setting every single day, bringing all this microbiology right back to the chair side. Let's talk viridans, streptococci. The sources call them commensals, but also opportunistic pathogens. What does that mean in practice?
This is where your microbiology knowledge becomes directly applicable to the majority of your daily practice dealing with oral health and disease. The viridans group Streptococci are a diverse collection of species that are mostly alpha-hemolytic, hence the name viridans from Latin viridis meaning green, referring to the hemolysis, or sometimes non-hemolytic. They are a major component, often the predominant component, of the normal bacterial flora of the healthy human oral cavity and upper respiratory tract. They colonize the mouth very early in life, often within hours or days of birth.
So they're normal residents, usually harmless or even beneficial.
In many cases, yes. As commensals, they occupy ecological niches and may help prevent colonization by more pathogenic bacteria. However, the opportunistic pathogen part is key. Under certain circumstances, particularly if they gain access to normally sterile sites, or if host defenses are compromised, these normally harmless bacteria can cause serious infections.
And their role is absolutely central to two major areas you'll deal with constantly as a dentist. First, let's talk dental caries again.
Absolutely. This cannot be emphasized enough. The sources clearly and consistently identify the mutans group of viridins Streptococci, particularly Streptococcus mutans, and its close relative Streptococcus sobrinus, as the major bacterial agents responsible for initiating dental caries, tooth decay. While caries is a complex process involving diet, host factors, and a polymicrobial biofilm, these specific bacteria are considered the primary drivers, the key culprits.
We touched on their virulence factors, acidogenicity, aciduricity, and polysaccharide production for plaque formation. Can we really hammer home how that translates directly into a cavity forming on a tooth step by step based on what the sources describe?
Yes, let's walk through that process because it's fundamental. It starts with diet, specifically the intake of fermentable carbohydrates, with sucrose, common table sugar, being the worst offender for S mutants.
Why is sucrose so bad?
Because S mutants and S sobrinus not only ferment sucrose rapidly to produce large amounts of organic acids, primarily lactic acid, that's their acidogenicity, but they also possess unique enzymes called glucosyltransferases, GTFs, and fructosyltransferases, FTFs. These enzymes use sucrose as a substrate to synthesize sticky extracellular polysaccharides, long chains of glucose, called glucans, like dextrin and mutin, and fructose, called fructans.
And those sticky polysaccharides are the glue for plaque.
Exactly. These sticky, often insoluble glucans are critical. They allow S mutans and S sobrinus to adhere tenaciously to the enamel surface, especially the acquired pellicle, and also to adhere to each other, building up the bulk and structure of the dental plaque biofilm. This biofilm isn't just a random collection of bacteria, it's an organized community embedded in this polysaccharide matrix.
So, the bacteria use sugar to make both acid and the glue that holds the plaque together on the tooth. What happens next?
This biofilm structure acts like a localized reaction chamber. It traps the acid produced by the bacterial fermentation right against the tooth surface, preventing saliva from easily washing it away or buffering it effectively. This leads to a significant and prolonged drop in pH at the enamel surface within the plaque, often falling below the critical pH level, around 5.5, at which enamel minerals start to dissolve.
So the plaque essentially holds a concentrated acid bath against the tooth enamel.
That's a good way to put it. Now, enamel normally undergoes natural cycles of demineralization, losing minerals when pH drops, and remineralization, gaining minerals back from saliva when pH rises. However, the sustained low pH environment created under a cariogenic plaque biofilm shifts this balance heavily towards demineralization. Calcium and phosphate ions are continuously leached out of the enamel's hydroxyapatite crystalline structure.
And the bacteria themselves can survive this acid bath they create.
Yes, and that's the other key virulence factor, aciduricity. S mutans and S sobrinus are remarkably tolerant of low pH. They can continue to metabolize sugars and produce acid even when the surrounding environment becomes quite acidic, conditions that would inhibit or kill many other less acid-tolerant oral bacteria. They essentially thrive in the acidic niche they create.
So they make acid, they survive the acid, and they keep making more acid, all while stuck to the tooth in their protective plaque. What's the end result?
Over time, this sustained acid attack leads to a net loss of mineral from the enamel. Initially, this might be a subsurface lesion, a white spot lesion, but if the process continues, the enamel structure eventually collapses, forming the clinically visible cavity or hole in the tooth. The sources also mentioned that high counts of S mutants in saliva are often correlated with increased caries activity or risk, and that the mutants group is also implicated as a major player in root caries, which occurs on exposed root surfaces when gums recede.
That step-by-step process, directly linking the specific traits of S mutants, using sugar, making acid, making plaque glue, surviving acid, to the demineralization process within the biofilm is absolutely fundamental. It is the scientific basis for so much of what you do in preventive dentistry: diet counseling, oral hygiene instruction, fluoride use, and restorative dentistry, removing decayed tissue and restoring the tooth.
Couldn't have said it better. Understanding that process is core knowledge.
Now, the other absolutely critical role of viridins streptococci in dentistry, one with potentially life-threatening consequences, infective endocarditis.
Yes, this connection is paramount clinical knowledge for every single dental professional, and the sources make this link very, very clear. Viridans group Streptococci, including species like Streptococcus sanguinis, Streptococcus mitis, Streptococcus oralis, and yes, even Streptococcus mutans are collectively the most common cause of subacute bacterial endocarditis, SBE.
SBE. This is a serious, often slowly progressing, but potentially fatal infection of the inner lining of the heart, particularly the heart valves. How on earth do bacteria that normally live relatively peacefully in the mouth end up causing such a devastating infection in the heart?
The key mechanism is transient bacteremia, the temporary presence of bacteria entering the bloodstream. The oral cavity, with its vast microbial population and rich blood supply, especially in the gingival tissues, is a frequent source of bacteria getting into the circulation.
How do they get into the bloodstream from the mouth?
The sources state clearly that bacteremia can occur frequently during routine dental procedures that involve manipulating the gingival tissues or the periapical region of teeth, or that cause bleeding. This includes procedures like tooth extractions, periodontal surgery, scaling and root planing, deep cleaning, placement of dental implants, and even endodontic root canal procedures where instruments might go slightly beyond the apex.
So invasive dental work is a major trigger. What about everyday activities?
Yes, significantly, the sources also point out that transient bacteremia can occur, albeit often with lower numbers of bacteria, during routine daily activities like vigorous chewing, toothbrushing, or flossing, particularly in individuals who have poor oral hygiene, gingivitis, gum inflammation, or periodontitis, more severe gum disease. The inflamed, ulcerated tissues in diseased gums provide an easier entry point for bacteria into the capillaries.
Wow. So bacteria from the dental plaque or the gingival sulcus get dislodged and pushed into the tiny blood vessels in the gums.
Exactly. Now, in most healthy individuals with healthy hearts, these transient bacteria entering the bloodstream are usually quickly recognized and cleared by the immune system, phagocytes in the blood, liver, and spleen, within minutes, causing no harm. But in patients who have certain pre-existing cardiac conditions that result in damaged or abnormal heart valve surfaces, or in patients who have artificial prosthetic heart valves or certain congenital heart defects, these circulating oral bacteria, particularly Viridans Streptococci, which are quite good at sticking, can adhere to these damaged or artificial endothelial surfaces within the heart.
They find a rough spot or a foreign surface like a prosthetic valve, and they manage to latch on before the immune system clears them.
Precisely. The damaged valve surface, often with little clots of platelets and fibrin already present, called non-bacterial thrombotic endocarditis, or NBTE, provides a receptive site for bacterial attachment. Viridans strep have surface adhesins that help them bind. Once attached, they can start to multiply and become coated with more platelets and fibrin, forming a protective structure called a vegetation.
A vegetation, like a little bacterial colony growing on the heart valve.
Exactly. It's a mass composed of bacteria, platelets, fibrin, and inflammatory cells. This vegetation can grow over time, physically damaging the heart valve and impairing its function, causing leakage or obstruction. Furthermore, the vegetation can continuously seed bacteria back into the bloodstream, leading to persistent fever and other systemic symptoms. Or, fragments of the vegetation, septigambuli, can break off and travel through the bloodstream to lodge in other organs, like the brain, causing stroke, kidneys, spleen, or extremities, causing infarcts or abscesses.
That sounds incredibly dangerous. And the subacute form caused by Viridans strep progresses more slowly.
Generally, yes. SBE caused by Viridans streptococci typically develops more insidiously over weeks to months, with symptoms like low-grade fever, fatigue, weight loss, night sweats, and heart murmurs, compared to acute bacterial endocarditis, which is often caused by more aggressive bacteria like Staphylococcus aureus and has a much more rapid and destructive course. But SBE is still very serious and can be fatal if not diagnosed and treated effectively with prolonged courses of antibiotics.
This entire pathway from oral bacteria entering the blood during dental work or even just brushing to them sticking to a susceptible heart valve and causing endocarditis is why the sources emphasize the critical importance of prophylactic antibiotics for certain patients.
This is a direct, potentially life-saving application of this microbiological knowledge that falls squarely on the shoulders of dental professionals. For patients who are identified through careful medical history taking as being at high risk of developing infective endocarditis based on specific underlying cardiac conditions, these conditions are clearly defined in periodically updated guidelines from organizations like the American Heart Association, administering a prophylactic dose of an appropriate antibiotic before undertaking certain dental procedures that are likely to cause significant bacteremia, like extractions, periodontal surgery, deep scaling, is recommended.
What's the goal of the prophylaxis?
The goal is to have a sufficient level of antibiotics circulating in the bloodstream at the time the bacteremia occurs, to kill off the susceptible oral Streptococci before they have a chance to adhere to the heart valves and establish an infection. It significantly reduces the risk of these oral bacteria seeding the heart.
So it's absolutely essential for dental professionals, dentists, and hygienists to be thoroughly knowledgeable about the current guidelines for endocarditis prophylaxis.
Absolutely non-negotiable. You must know which specific cardiac conditions put a patient at high risk, which dental procedures necessitate prophylaxis for these patients, and which antibiotic regimens, drug, dose, timing are currently recommended, including alternatives for patients with allergies. Following these guidelines is a direct professional responsibility to prevent this severe, potentially lethal complication originating from the oral cavity. It's one of those direct links between dental practice and saving lives.
That makes the connection incredibly clear and highlights a really critical aspect of safe patient care in dentistry. Okay, beyond the huge topics of caries and endocarditis involving viridin strep, what other oral or head and neck infections involve streptococci that dental students need to be aware of?
The sources specifically mention the anginosus group, S. anginosus, S. constellatus, S. intermedius, also known as the S. milleri group. These are recognized as important pathogens in pyogenic pus-forming infections, particularly in the head and neck region. They are frequently isolated from dental alveolar abscesses originating from teeth, infections following dental procedures, and also from more serious deep neck space infections, brain abscesses, and liver abscesses, often as part of a mixed infection. A key feature noted is that they are often microaerophilic or capnophilic, meaning they prefer reduced oxygen and organism increased carbon dioxide for growth, which can make them trickier to culture sometimes.
So the anginosus milleri group are major players in dental abscesses and related pus-forming infections. What about the anaerobic strep?
Yes, the anaerobic streptococci, many of which were formerly classified in the genus Streptococcus, are also commonly implicated in polymicrobial anaerobic infections originating from the oral cavity. The sources mention their role in infections like pericoronitis, inflammation and infection of the gum tissue overlying a partially erupted tooth, usually a wisdom tooth, and other dentoalveolar abscesses, typically working synergistically with other anaerobic bacteria like Fusobacterium, Prevotella, or Porphyromonas.
So often part of a mixed bag of anaerobic bugs in those types of infections.
Exactly. And of course we need to revisit Enterococcus faecalis in this context. While not a typical primary cause of initial endodontic infections, its significance, as highlighted, is in persistent or secondary endodontic infections. That is, root canals of teeth that have failed initial treatment or become reinfected. Its remarkable ability to survive the challenging environment inside a treated root canal, invade dental tubules, form biofilms, and resist common antimicrobial treatments makes it a key target in endodontic retreatment cases.
Right, the survivor bug in endo. So understanding which specific groups of Streptococcus mutans for caries, viridins for endocarditis risk, anginosus and anaerobes for abscesses, and Enterococcus for endo failures are commonly associated with specific oral infections really helps guide diagnostic thinking and potential treatment decisions, including choosing the most appropriate antibiotics if needed.
Precisely. And all of this feeds back into that broader concept mentioned briefly in the sources. The oral systemic axis, or oral systemic link. The mouth isn't isolated like Las Vegas. What happens in the mouth doesn't always stay in the mouth. Oral infections, the inflammatory burden from conditions like periodontitis, and even routine dental activities can lead to bacteremia and the dissemination of oral bacteria and inflammatory mediators throughout the body. This can potentially contribute to or exacerbate systemic diseases beyond just endocarditis, although endocarditis remains the most direct, well-established, and life-threatening link for oral streptococci specifically. But the connection is broader than just that one disease.
Given this constant presence of potentially pathogenic microbes, like various streptococci in the mouth, and their proven ability to cause local disease and access the bloodstream, this really puts a huge spotlight back on the importance of those everyday procedures in the dental office. Infection control.
It absolutely does. The ubiquitous nature of streptococci and the entire complex oral microbiome is the fundamental biological reason why rigorous, meticulous infection control protocols are absolutely non-negotiable in any dental practice setting. The sources explicitly list the essential pillars of standard precautions. First, using appropriate personal protective equipment, PPE, gloves for every patient contact, masks to protect mucous membranes from splashes and aerosols, protective eyewear or face shields for the same reason, and sometimes gowns or lab coats.
Check. PPE is essential.
Second, meticulous hand hygiene. This means washing hands thoroughly with soap and water, or using an alcohol-based hand rub before putting on gloves, after taking gloves off, and between patient contacts. It's one of the simplest yet most effective ways to prevent cross-contamination.
Critical. Hand hygiene.
Third, the proper sterilization of all reusable dental instruments that penetrate soft tissue or bone, critical items, or contact mucous membranes or non-intact skin, semi-critical items. This typically involves cleaning followed by heat sterilization, usually in an autoclave, to kill all microorganisms, including bacterial spores. For items that can't tolerate heat, high-level disinfection might be used.
Sterilization is key for instruments.
And fourth, proper disinfection of environmental surfaces in the treatment area between patients using appropriate EPA-registered hospital disinfectants, especially for surfaces that might become contaminated with blood or saliva.
Surface disinfection. Right. These aren't just bureaucratic hurdles or optional extras. They are direct evidence-based defenses against the transmission of organisms like the Streptococcus species we've been discussing all this time. They prevent patient-to-patient transmission, patient-to-staff transmission, and even staff-to-patient transmission.
Precisely. Every step in that infection control chain is informed by our understanding of microbiology, including the potential for organisms like Streptococcus, which are present in virtually every patient's mouth, to be transmitted and cause harm if these barriers break down. And the sources even briefly touch upon another aspect of infection control relevant to the dental environment: managing dental unit water lines, DUWLs.
Yes, the water that comes out of the air water syringe and handpieces.
Exactly. Those long, narrow tubes can easily develop biofilms formed by environmental bacteria, including some opportunistic pathogens. While not typically involving pathogenic Streptococcus, managing DUWLs through regular flushing, disinfection protocols, and using sterile water for surgical procedures is another important part of maintaining a safe treatment environment and preventing potential exposure to waterborne microbes. It all connects back to controlling microbial risks.
Absolutely. So we've really taken a comprehensive deep dive into the world of Streptococcus today. We've covered its basic features: gram-positive cocci, often in chains, catalase negative. We explored the various ways we classify its incredibly diverse species using hemolysis, Lancefield antigens, biochemical tests, and molecular methods. We unpacked the potent tools, the virulence factors like M protein capsules, spreading enzymes, superantigens, toxins, and biofilm formation that different species use to interact with us and cause disease.
And we traced the wide range of diseases they cause, from systemic infections like strep throat, pneumonia, meningitis, and toxic shock syndrome, and then focused intently on those hitting close to home in dentistry. S. mutans driving dental caries, viridin Streptococcus posing the major risk for subacute bacterial endocarditis linked to oral bacteremia, and other groups like anginosis, anaerobic strep, and Enterococcus faecalis playing key roles in dental abscesses and endodontic infections.
We also looked at how clinical labs identify these microbes using microscopy, culture, biochemical tests, and antigen detection. And importantly, we discussed the principles of treating streptococcal infections with antibiotics, noting the critical issue of emerging resistance and the paramount importance of prevention strategies in dentistry, especially antibiotic prophylaxis for endocarditis in high-risk patients and rigorous daily infection control.
You've really seen how these seemingly simple gram-positive cocci are actually incredibly complex, adaptable, and significant players in human health and disease. They drive some of the most common oral diseases you will treat every single day, and they pose significant risks for serious, even life-threatening systemic complications that you, as a future dental professional, are uniquely positioned to help prevent.
Understanding Streptococcus and all this detail isn't just about passing your microbiology exams. It's truly fundamental to practicing safe, effective, and comprehensive patient-centered care throughout your entire dental career. This deep dive into the sources really cements that undeniable connection between basic science and clinical practice.
It absolutely does. And reflecting on all this, it naturally leads to an important question for you to continue pondering as you progress through your dental education and prepare for the realities of practice. Considering the ever-increasing global challenge of antibiotic resistance, which affects various streptococcal species, including those commonly found in the oral cavity like Enterococcus and certain viridins groups, and given the critical importance of using antibiotic prophylaxis appropriately to prevent devastating infections like endocarditis, how might the evolving landscape of microbial resistance, combined with our deepening understanding of the complex oral microbiome, impact the current guidelines and shape future strategies for preventing and managing streptococcal infections in your vulnerable dental patients? How will you navigate that balance between necessary treatment and responsible antibiotic stewardship.
That's a really crucial question to think about. Balancing the need to treat and prevent infection with the need to preserve antibiotic efficacy for the future. That's going to be a major challenge and responsibility for your generation of dental professionals. Something to definitely keep in mind.